Welcome to our webinar entitled Breakthrough in Machine Learning from Small Data Sets, including Designed Experiments. Our agenda today, we're going to start with some introductions, and then we'll talk about the what, the why, and the how of S-VEM. That's the central topic of today, and we'll do a quick overview of DOE and machine learning and describe blending them into S-VEM, analyze real-world experiments, and review some current research in this area. We'll follow that with a demonstration of our S-VEM product. And finally, we'll do next steps and Q&A. So today you will learn about S-VEM, a remarkable new method to extract more insights and build more accurate predictive models from small sets of data. So we like to think about our brains. They are great knowledge creators. And that's what S-VEM, or about what we do with modeling. We are generating knowledge. We're creating knowledge. So these methods and tools like JUMP are very helpful in leveraging our thinking and accelerating our thinking. But at Predictum, we also think that the brain is a really lousy knowledge container. So if I may, I'd like to just mention briefly a couple of other products in this area. Sash Lab, which is about formulations and compounding, and Cobase, which is for general research, experimentation, problem solving, improvement initiatives. Both of these capture explicit knowledge, the models that are created, if you will, and their underlying data, and make them available for reuse by others. So this way we're able to avoid delays, and you can avoid costs associated with regenerating knowledge that others have created in your company. I mean, just think about it. When people are not available or when they leave the company, you want that knowledge to stay and be accessible to others. And that's what these products do. I'll leave it to you to go to our website and look them up there. But our focus is on the other side, which is generating knowledge. And my name, just to introduce the speakers, is Wayne Levin. I'm the president of Predictum. And Chris Gotwalt is the director of JUMP Statistical R&D. And joining us as well is Philip Ramsey. He's a senior data scientist at Predictum and also a professor at the University of New Hampshire. I do want to recognize JUMP's many contributions to data science. Just in the DOE area alone, going back about 20 years, we see the coordinate exchange algorithm, which made possible optimal designs, which plays a role in S-VEM, in fact. Also definitive screening designs. Both of these were led by Dr. Bradley Jones, and it's Dr. Chris Gotwalt who, as I mentioned, brought us S-VEM. So we're just one of a number of things, again, that JUMP has brought to industry. So what is S-VEM? You've heard me say it a number of times. Well, it stands for Self-Validated Ensemble Models. And what it does is it combines two well-practiced methods, machine learning and designed experiments or design of experiments. And it overcomes the limitations of limited amounts of data, like what you find in DOE. So S-VEM works exceptionally well. We have been using it at Predictum with our client data in semiconductor and pharmaceutical and in chemical industries, and we have been super impressed. We really think that this method has surpassed what we've seen in conventional DOEs. And we've been using it where the number of runs is less than the number of parameters. So in other words, super saturated situations. But it does better than other methods in super saturated designs. We were able to fit higher order models, which you don't typically find in designed experiments. We get more accurate predictions. And we've also been able to recover from troubled DOEs where there was a lack of power, where otherwise you'd have to augment the design and add more runs and spend more money and delay realizing your objectives. We've been able to use S-VEM in these cases instead of adding runs. Also, when you have questionable or missing runs, S-VEM has been very helpful in that area. And so what I'd like to do now is pass it over to Chris, who's going to talk about the what, the why, and the how of S-VEM.
Thanks, Wayne. Now I'm going to give an introduction to S-VEM go over a case study that illustrates how it works on some real data, and then talk about some of the simulations that Trent Lemkis at the University of New Hampshire has done that gives us the reasons why we think this is such a potentially revolutionary technique. So now I'm going to introduce the basic idea of self-validating ensemble modeling, or S-VEM. I'll try to keep it fairly simple, and I'll assume that you have some experience of design and analysis of experiments. Most importantly, you should understand that the statistical modeling process boils down to fitting functions to data that predict a response variable as a function of some input variables. At a high level, S-VEM is a bridge between machine learning algorithms used in big data applications and design of experiments. Machine learning and DOE each have a long history, but until now they were somewhat separate traditions. Our research indicates that you can get more accurate models using fewer observations using S-VEM. This means that product and process improvement can happen in less time using fewer resources. So if you're in a competitive business environment where being first to market is critical, then you may want to hear what we have to say. We believe that S-VEM has tremendous promise in the pharma and biotech industries, high technology like semiconductor manufacturing, and in the development of consumer products. In my role as a research and development director for JUMP, I've had something of a bird's eye view of how data is handled in a lot of industries. I've developed algorithms for constructing optimal designed experiments, as well as related analytical procedures like mixed models and generalized linear models. On the other hand, I'm also the developer of JUMP Pro's neural network algorithms and some of the other machine learning algorithms that are in the product. Over the last 20 years, I've seen machine learning become more prominent when modeling large observational data sets. And it's been fun watching how new algorithms have been developed, and we've had tremendous progress made in this area of modeling with observational data. At the same time, the analysis of designed experiments has also changed incrementally over the last 20 years. However, the statistical methodology of industrial experimentation would be recognizable at this point to anyone that read Box Hunter and Hunter in the 1970s. Machine learning and design of experiments are generally applied in industry to solve problems that are predictive in nature. Is the sensor data indicating a faulty batch? Would be a question that would be answered with a machine learning algorithm. How do we tweak the product so we can maximize yield while minimizing cost? Is a problem that would be solved using design of experiments. Although machine learning and DOE are both predictive in nature, they are often applied to rather different types of data. What I'm going to do now is give a quick overview of both methodologies, and then I'll describe how we've blended them into S-VEM. I'll analyze data from real experiments to show how it works and why we think it's doing very well. Then I'll go over the results of simulations done by Trent Lemkis at the University of New Hampshire and discuss open questions and next steps in the research that needs to be followed up on. In machine learning, what we want to do is we want to predict a response variable as a function of input variables. So in the example before you, we see some real data from a manufacturing process where the response of interest was product shrinkage at the end of the manufacturing step, and some information that was collected during that step. We want to identify a function of compaction pressure, sintering time, and sintering temperature that predicts this shrinkage. So in the simplest form of machine learning type modeling, what we would do is break the data up into two sets. A training set that would be used to fit a variety of models like neural networks, linear regressions, support vector machines, random forests. And then we also have another set of rows that is called the validation set that we use to assess how well those models are each going to generalize to new data sets. So in DOE modeling, the problem is very similar. So you might want to model response, such as shrinkage again, as a function of some dials that you can turn on your manufacturing process or in the way that you set up your product. So the difference is that we're usually setting the X's explicitly as part of the design of experiments methodology, changing them very carefully and explicitly, and then going through a different set of analysis procedures that are geared towards small data sets in order to be able to get the most from the small number of observations that we have. These are things like effect tests and linear models, half normal plots, and even more recently developed techniques like using forward selection with the AICC model selection criteria to identify which factors are the important ones and which interactions need to be kept. But we don't usually apply the same kind of partitioning of the data into training and validation when we're doing DOEs because we just don't have enough rows. If we were to do that, the software will often tell you that you've induced singularities into your model and you're not able to fit the models that you had originally intended to. Now, as a thought experiment, one might ask the question, well, can't we cheat a little bit? Can we go ahead and maybe take the data set, double it up so that we have all the rows from the original DOE as training and all the rows from the original data as validation? Now, this idea is a pretty bad one. It's crazy because when we do that, we now no longer have any independent assessment of goodness of model fit. So every time we make the model more complicated and fit it to the training set, we are by definition also improving our fit on the validation set, and will always overfit the data, choosing the most complicated one of all the options. A useful way of thinking about the machine learning modeling process is to get away from thinking about it in terms of assigning rows into training or validation subsets and thinking about it instead as having a training weight column and a validation weight column, so that rows where the training weight is equal to 1 are the training set, and the rows where the validation weight is equal to 1 is the validation set. When we plot the training and validation weights, we see that they are perfectly anti-correlated. Now, remember that the problem with applying machine learning to design experiments is that we often cannot afford to hold out entire observations and put them into the validation set. What if there was a way to create fractional weights that were anti-correlated, similar to these, but had fractional values so that we were able to have partial observations contributing to training and parts of observations contributing to validation, and those weights are anti-correlated so that when an observation contributes more to training, it contributes less to validation and vice versa. Well, it turns out that there's a very simple way of doing that. A straightforward way to create anti-correlated weight pairs is to first create a uniform random variable between 0 and 1 and run that variable through the exponential distribution's quantile function. That will create a training weight that is exponentially distributed with a mean of 1 and a variance of 1. Then, now, if u is uniform distributed, then 1 minus u is also uniform distributed, and it's perfectly anti-correlated with the original u. We can run 1 minus u then through the exponential quantile function, and we will get another exponential random variable that is maximally anti-correlated with the original one that we'd created. So if we use this trick to create a couple hundred weight pairs, we would see a sample of data that looks kind of like what we see before us. The training weights and validation weights are both identically exponentially distributed, but if we plot them on a scatter plot, we see that they are strongly anti-correlated with one another. It's not possible to make them perfectly anti-correlated because both of them have to be positive because of the exponential distribution. But this is about as good as we can get. So when we're modeling with S-VEM, what we do is we have our original data, our input variables, and our responses. We create our training and validation weight pairs. Then we fit some statistical model to the weighted data and save its prediction formula. This could be a model of almost any kind. So it could be a linear model, it could be a response surface model, a support vector machine, a neural network, it could be kind of anything. Then we reinitialize the weights and train and tune another model and save its prediction formula. And then we do that again and again and again, probably a couple hundred or 1,000 times And then when we're done, our final prediction formula is the average of all the what we call out of validation reps of the models that we had fit over this process. So let's apply this to the modeling of the shrinkage response. So here we have our input columns over to the left, followed by shrinkage. We have the uniform random variable, the training weight, and the validation weight. Notice how the training and validation weights are anti-correlated, so that when the training weight is large, the validation weight is small, and vice versa. So then we would go ahead and we'd fit a model to shrinkage, save the prediction formula. If we took a look at it, in this case we're using forward selection, and it shows compaction pressure and sintering temp as the variables it wanted to keep. Then we update the weights, save another prediction formula, and this time a different set of main effects and interactions was kept by the auto-validation process. Then we do it again and again and again. Ultimately, we might do this 250 times. We create a new formula that's the average across all of our auto-validated forward-selected models. And we have our S-VEM prediction model, which we can then go into the jump profiler to use to actually optimize our process. So now I'm going to introduce some data that I'm going to use as a proof of concept for S-VEM, the plasmid DNA data. So this is data that was created some time ago around the fermentation step of a Pdna manufacturing process. And this data was collected in collaboration with Phil Ramsey about 10 years ago, when definitive screening designs were a new type of design that had just come out, had been invented by my colleague Bradley Jones. And Phil Ramsey was interested in seeing how well the DSD model performed relative to a classical design, a central composite design. And so when he was working with the experimenters, he had them do two separate experiments with separate people running the experiment using different test equipment. Both experiments had the same factors: pH of the fermentation solution, percentages of dissolved oxygen, temperature for the fermentation media, the microbial mass of E. coli in units of optimal density at 600 nanometers, the feed rate, which is a glucose measure, and then our response was the total amount of Pdna that was coming out in the titer. Here we see the data all put together in a jump data table. What we're going to do is we're going to hide and exclude the rows that corresponded to the central composite design. We're going to use that as an independent test set after fitting several different models to the definitive screening design rows in the data set. This way we can see how well some classical procedures perform on the central composite design's observations, and also S-VEM. So now that we have it set up, we're just going to go ahead and run the script, which executes S-VEM by applying forward selection 250 times, and then we can go ahead and do a couple manipulations just to clean up the columns and create our S-VEM average formula. We can also set up the standard deviation, and I'm just going to go ahead and group the columns and hide the AV rep formulas just so that things are a little bit easier to read. So now I've got my AV rep columns hidden. We're going to use the average across them as our overall model for S-VEM. And then we're also going to take the column-wise standard deviation across those to characterize the uncertainty in our predictions. So I've named the second column Pred SE so that the profiler platform will know that it's a standard error formula. So I get those loaded up as responses, tell it to expand intermediate formulas, confirm that it's a standard error formula, and we see the traces of titer as a function of the input variables now. I'll do another simulation where I randomly sample the input variables to do a assessment of variable importance. We see that feed rate is the most important at 81% of the response surfaces variation explained by feed rate, followed by dissolved oxygen at about 9%, and then the remaining variables are at around 8%, 3%, and 2% respectively. It's very quick, so if we wanted to optimize this process, it's very clear that adding glucose is beneficial to yield. Dissolved oxygen has a slightly decreasing effect, as does pH, and then the other two variables, it looks like it's fairly flat in those. So we can set those to convenient settings if we'd like to. So prior to running S-VEM, I used the FitDSD platform to find a model for the observations in the DSD. And then I also went into the generalized regression platform and used forward selection in combination with the AICC model selection criteria to find a model also. So what I'm going to do now is I'm going to go ahead and unexclude the central composite design rows, and then go into the model comparison platform to see how these three models are performing on the CCD runs. I'm going to use experiment as a grouping variable, and we have to let it know that titer is the response. So what we see is that S-VEM has an R-square on the central composite design runs of 55%. So these are runs that were not used to fit this model at all, and it's got S-VEM is giving us a much higher predictive capacity than either forward selection with the AICC or the FitDSD approach to model selection. Those both have about half the R-square that the S-VEM model does. This is pretty remarkable and is fairly typical also of the kinds of accuracy that we're seeing on examples that we try S-VEM out on in conjunction with other variable selection approaches that have been around for longer. Taking another look at the results of these three models, we see actual by predicted plots. This is the FitDSD prediction model, followed by forward selection, followed by S-VEM. On actual by predicted plots, we want to see the data on independent test data like this as tight as possible along the 45-degree line, and it's pretty clear that S-VEM here is tighter along that 45-degree line than the other modeling approaches that we tried this out on. So now I'm going to try something a little bit adventurous. I'm going to take the complete data set with all the DSD and CCD runs together, and I'm going to fit an S-VEM neural network to it. This way we can see how close the other classical approaches and the S-VEM model that was fit to the DSD are at recovering the shape of a sophisticated model that is being fit to all the rows in the data set. So I go ahead and get this set up, and we will go to the profiler and take a look. So now we have the profiler of the three models that were fit to the DSD and then the neural network that was fit to the combined data set. And we see that the S-VEM model fit to the DSD looks the most alike to the neural network that was fit to the complete data set. We also see this interesting feature here where the feed rate impact seems to flatten off at about 3.0, which apparently makes some sense based on the biology. So if you give a lot of glucose to some bacteria or a fungus, there's only, it can only grow so much. If you can have too much, it only is linear for so long. And this neural network is capturing that. So the Pdna experiment is pretty encouraging for S-VEM, but it's only really an N of 1. So now I'm going to go over some simulations that were done by our graduate student at University of New Hampshire, Trent Lemkis, where he's done simulation studies comparing S-VEM to other classical procedures and other different ways of tweaking auto-validation methods in general, where we looked at definitive screening designs and box banking designs in 4 and 8 factors. We used 1,000 simulation reps per situation. Each simulation rep had its own set of true model coefficients. The non-zero ones were exponentially distributed with a mean of 1. The error variance was 1. And the true coefficients represented 50% to 100% of all possible coefficients. So this was part of the design of the simulations as we looked at how accurate the different modeling strategies were at different levels of saturation and supersaturation in the parameter space. Of all the methods that we tried, we compared them on an independent test set, root mean squared error. This was calculated using a 10,000 point space filling design, and these RMSEs were calculated versus a true model. There was a whole zoo of different methods that we tried, but in the end, we can tell the story pretty simply with this graph right here. So this is just one of the simulation setups. It was on the definitive screening design in 8 factors, and 50% of the factors were active. So now note that this design had 21 runs and about 21 or 22 active factors. So it is a saturated model. And the independent test set RMSE for the S-VEM methods, which we're calling here by AV6, it was our prototype name for what became S-VEM. This RMSE on the test set is much lower than other classical procedures such as the lasso with AICC, Danzig selector, forward selection, prune forward selection, or even the best of the fit definitive screening type approaches in and so S-VEM here is doing substantially better than the other approaches because it's more accurate. Here's a similar set of situations where everything is the same, except now we are allowing all 45 design matrix parameters. All the regression coefficients are exponentially distributed with a mean of 1. Now, we still only have 21 runs, so effect sparsity is now false. We are fitting models accurately using S-VEM type methods that are still doing pretty well, whereas the classical approaches are doing somewhat worse. And so it was this plot when we were doing our research with Trent this summer where I became kind of a believer in this whole approach. I never thought it would be possible to fit models to data of designed experiment type data where you had more active effects than there were runs. And this is working. It's really, really remarkable. We've kind of broken the speed of sound in statistics by having models with more parameters than there are runs accurately fit to data. So we're very excited about what we've been seeing from S-VEM so far. We believe that it could really change the way that industrial experimentation happens in the world. So, for one thing, it changes potentially the emphasis from a p-value based mindset when we're designing and analyzing experiments to a more predictive modeling type way of looking and thinking about things. I will say that I do think that hypothesis testing and so forth will still play a role in confirmatory studies or anything where you're trying to establish a treatment effect in a clinical trials type situation, I think that p-values will still be important there for some time. But when you're internally optimizing processes and making products better, I don't know what the value is anymore of having p-values. We still have quite a lot of questions that we don't know the answer to yet. One thing I showed as a part of the demo here today is using the column standard deviation as an uncertainty estimate for the predictions. There are other ways that we will be investigating to characterize prediction uncertainty, such as bootstrap quantile type intervals. So far I've been showing mostly quadratic RSM models and a little bit of neural networks. But this really opens the door to a lot of other different kinds of base models. There is the Cornell-Montgomery framework that has quadratic interactions. There are different kinds of neural networks. There's regression trees, support vector machines, different kinds of neural networks with different base architectures. Which ones of these are going to be the ones that will make our accuracy the highest? It also opens lots of questions about what kinds of designs do we use. I think that there's a real opportunity here for Bayesian eye-optimal designs to see an increase in their usage because they allow for supersaturation while at the same time maintain a prediction orientation. I think we have to reconsider what the role of screening experiments and sequential experimentation. We don't know. It may be easier just to go ahead and go for a response surface model with curvature and interactions rather than doing a round of screening, now that degrees of freedom don't really cost us that much anymore. And actually, speaking of degrees of freedom, what does this say about power and sample size considerations? It's probably the case that power and sample size calculations are very pessimistic when you start applying S-VEM. And so there would need to be new ways of determining what the meaning of power is in a predictive context and also how to do sample size determination prior to experimentation. So far, also, S-VEM is making very few assumptions about the data. The most important one is that the rows are independent. We're not really assuming normality. We're using an empirical bootstrapping type inferential framework and estimation framework. And so as long as the data are IID, we're pretty good. Now, when you get into blocked experiments, split plot experiments, we have ideas for generalizing S-VEM for these situations. And then also when you get into measurement system studies where you've got multiple variance components, we'll have to be doing some thinking about how we can generalize S-VEM to that, because the way that we're generating the simulation weights are pretty much assuming that you've got independence across rows, and that's not the case for those kinds of studies. So now I'm going to hand you back over to Phil, and he is going to show you more about how to apply S-VEM on your own data.
Thank you, Chris, for that nice discussion of the S-VEM methodology and the concepts behind it. In this section, I'm going to continue on with the case study Chris mentioned, that is the plasmid or Pdna fermentation process experiment. And we know with the rise of gene and cell therapies, Pdna is becoming more and more important to biologics manufacturing and delivery. So recall, the original experiment is a 5-factor, 15-run definitive screening design. And the response is the titer of Pdna. But also remember, completely separate, and it's important to note this is completely separate, a 31-run, much larger central composite design was performed, and the goal was to compare the two designs and see if the much smaller definitive screening design would reach the same conclusions as the 31-run design. And again, our entire goal here is to develop models which accurately predicts the titer over the entire design space, not just finding an optimum, but also characterization. So at this point, I'm going to go ahead and switch over to jump. And what I want to do is demonstrate a product we have developed at Predictum, which is built on the Jump Pro platform. And what the product does, it more or less automates the S-VEM procedure. So I have installed the product. So under the Predictum tab, I am going to select self-validating ensemble modeling. Okay. In the background, the auto-validation set has been created. You can access if you want to, but it is not necessary. And then what we have to do is define a model. In this particular case, I'm going to use an unusually large model. This is an extension of what many people know of as the full quadratic model, a model often used for optimization. It contains the main effects, two-way interactions, and quadratics. What isn't well known is that model may be good at finding an optimum. Often it is not, its performance is not very good for characterizing an entire design space. The model simply cannot account for the nonlinear behavior that we see across the design space. So what I'm going to do, I'm just going to add a model. In this model, I sometimes call the Augmented Full Quadratic Model. This is actually based on a wonderful paper by John Cornell and Doug Montgomery from 1996. And what they do, they take the full quadratic model and they augment it with a number of higher order terms, many of which are linear by quadratic interactions. And I'll just give you an example. Right there, here is an interaction of the pH squared with induction temperature. And what these terms do, they account for compound curvature, which is typically the type of behavior that you see in a real chemical or biological system. The full quadratic model itself cannot account for this changing amounts of curvature as you move about the space. And our response is a titer of Pdna in milligrams per liter. So I'm going to click on run. It'll open up GenReg. Okay. So notice you have a number of options in here. We won't get into all of them, including estimation method. I'm going to just leave it at the default less O. And how many iterations do we want to run? Well, it'll take up to 10 or 15 minutes if I do, for instance, thousands of iterations. So for convenience of demonstrating the product, I'm just going to do 20, a small number. In reality, we would typically do hundreds and in many cases, thousands of simulation runs. So I click on go. So remember, the auto-validation set already exists in the background. Okay. And we're done. So what we get as output is an actual by predicted plot. So it looks like we have a pretty good fit. And I want to point something interesting out, and this is a subtlety. That augmented full quadratic model had 40 potential predictors. I only have 15 observations in my design. So in classic DOE modeling, one could not possibly fit a 40-term model. But using these concepts from machine learning, and that's where ensemble modeling comes from in the S-VEM term, I actually can fit a 40-term model. And as I'm going to show you in a moment, the actual 40-term model I fitted over about 2,500 bootstrap runs actually fits well. So what do I do with this model? Well, what we would do By the way, again, from the actual by predicted plot, it seems to fit the DSD data quite well. And I see nothing of real concern in the residual plot. So under the self-validating ensemble modeling menu, select Save Prediction Formula. Now I'm not going to do it here because I already have saved the formula. So we come back to Jump. And notice it says Prediction Formula. And this is the entire 40-term model. And it's been estimated using model averaging, again, over 2,500 boot runs. And what I'm going to do is take this formula and I'm going to actually deploy it to the CCD data. Okay. So remember, the CCD, I'll just show you quickly, was a companion experiment. Again, I can't emphasize enough that these were run separately, separate batches of raw materials. A lot of the fermenters were different, operators were different. So this was very much a different experiment. What I'm doing here, I'm using the CCD as a true validation set. So I fit the model. I've deployed the model to the data set containing the CCD data. And what I'm interested in seeing is, well, how well did our DSD model perform? So if the behavior of this process is similar in the DSD and the CCD, then when I use the model on the CCD data, remember the CCD data, Central Composite Design data, was not used at all in fitting the model. So in that sense, we say it is a true validation set. So what I'm going to do is just show you an actual by predicted plot. Okay. So what you're looking at, and this is only on the 31 CCD runs, the x-axis is the predicted Pdna. This is using the DSD formula. Remember, the DSD is half the size of this run, okay? And then the y-axis is the actual measured Pdna. So as you can see from the actual by predicted plot, okay, the actual lead model does a good job of predicting the Pdna results in the CCD, indicating that the process performance, in other words, the relationship between the factors and the response, is similar in the CCD to the DSD, or otherwise this would be just a random scatter plot. To help you see better the trend, I've actually put prediction intervals on each side of the line. And that's there, frankly, just to illustrate to you the clear linear trend. Remember, in an actual by predicted plot, if your model is predicting accurately, then this model should have a linear trend with the points randomly distributed about the diagonal line. So you can see this is actually a pretty good fit. In fact, I'll do one other thing for you. I can show you right here. Okay. The actual prediction standard deviation, we call it the root mean square error of prediction, is about 67. Now that's actually, I mean, it may seem high to some of you, but that's actually pretty good. Again, you have to understand that this CCD is completely independent data, and we used auto-validation and ensemble modeling on a 15-run DSD. In other words, we saved a lot of time and effort and money running the smaller design. And we built a model, and indeed that model predicts very well. So the idea, again, of this demonstration was to illustrate to you how the Predictum product, the S-VEM product, works. And again, you can contact Predictum, for instance, Wayne at predictum.com, and he'd be happy to talk to you about the S-VEM product. And we anticipate over time this method is going to grow in popularity, and we'll be adding features as we go. But right now, it functions very nicely in performing S-VEM. Thank you. And Wayne, I'll turn it over to you.
Thank you very much for that demonstration, Phil. And speaking of Phil, Phil and I are producing an on-demand course that you can take online on mixture designed experiments, and they will include S-VEM. Another announcement is that the S-VEM product, which takes the form of a jump add-in, is available for your evaluation. So that would be our next steps. There's basically two ways that you can try S-VEM yourself. One is just try the product, and if you send me an email, you'll need Jump Pro in order to do this, version 14.3 or 15, 15.2 being the latest. Now, if you don't have Jump Pro, you can get a trial by way of this link. And if you look below, you'll see there's a link for downloading the slides. So you can click on the link here once you've downloaded that, and you can try this out for 30 days. The other way you can do it is contact us at Predictum, and we can work with you on your historical DOEs, run it as a proof of concept, do some comparative analysis. We can do this on a limited basis. And of course, we'll review the analysis with you. So that takes us to the end here. And I'd like to open it up for some Q&A. And down at the bottom again, for evaluation of S-VEM, you can send an email to myself or to info@predictum.com.
Hello, everyone. So there's been quite a few questions that have come in, and I'd like to take some time to answer some that have come in through the chat. Please continue to ask questions in the chat, and we'll answer them as we go. So taking a look at some of these here. So I want to point out that this webinar is being recorded, and if you want to email Wayne at Levin at predictum.com, he'd be happy to send you a link to the recording once we have that prepared. One of the questions was, let me go back to the beginning. Yes, so one of the questions was, wasn't this presented at the Discovery Summit last week? And yes, there was some material presented. Phil and Tiffany Rao presented on it was called auto validation at the time. So if you just look up Phil Ramsey at the Jump Discovery America's 2020, you'll be able to find a link to that that presentation. There was another question about the standard error of the ensemble model that I showed. So what we did was I saved out a bunch of prediction formulas. So maybe like 200 prediction formulas as part of the S-VEM process. The final model is the mean of those. And we just take the column-wise standard deviation of those observations to calculate a standard error. It's the same kind of thing as a quantile-based, it's calculating a bootstrap standard error and applying that to your estimate. Only we're using formulas rather than point-wise estimates for this. So when we're creating the data, one of the questions also was, how much more data can you create with the weighting? If the original data had n rows, how many times can you fold the initial x rows to create validation and test sets? So we're re-initializing the weights hundreds, if not thousands of times when we're doing S-VEM. So we're creating a bunch of fold pairs for each of those. I have ideas for how it could be broken up into 3 sets, but that hasn't really been evaluated yet. So we're not really ready to talk about it. We're actively, this has opened a whole huge can of worms for us. And there's just many great ideas that we're pursuing. And so just kind of stay tuned for that. how can you assure that none of this is overfitting? Well, I think that the important thing here is that you would still want to run validation runs or run checkpoints at the end to validate the predictions. I don't think that there will be ever a true surrogate for replication, and so you would still want to determine whether or not the model is any good, just like you would with any other model in any other context. There was a question about comparing S-VEM models to PLS. So S-VEM is like a general model creating machine that you can insert pretty much any modeling strategy into. And so you could apply S-VEM to partial least squares models and perhaps improve PLS the same way that we're seeing improvements in the fitting of lasso and forward selection type models to designed experiments. PLS is usually only really covering linear terms and maybe some interactions and curvatures, but S-VEM models, we've been exploring a lot of these partial cubics or Cornell-Montgomery type extensions of the response surface model to include interactions of main effects with quadratic terms. And I haven't ever seen that applied in a partial least squares framework yet.
Hey, Chris?
Yes, Phil?
I'd add to that, with partial least squares, it primarily uses k-fold cross-validation, which you obviously can't do with data like designed experiments that are highly structured. You can't really do it well with observational data with things like rare events. So in those cases, I have looked at partial least squares informally using the S-VEM approach, and it actually works very well. In other words, I was able to generate models and I had a small validation set I had, and they predicted well. But as you said, this just opens up so many opportunities, and what's needed are people to get on board and consider doing some of their own research. But I think it's going to actually revolutionize partial least squares, because I'm very skeptical of the k-fold strategies that are used. And oh, by the way, a lot of times in PLS, you have more factors like in spectroscopy than you have observations. It's super saturated. So I think it's an interesting question, and I think PLS could be reinvented with S-VEM. That's all I had to say on the subject.
All right. There was another important question that came in asking about how well these models are performing outside the design space. My experience has been that because we're doing model averaging, the performance outside the design space with mild extrapolation is actually better than the standard techniques. So let me see if I can share my screen. I can show you the, so this is the design that we were just talking about with for the Pdna data. So the points in blue here, this is a scatter plot matrix of the factors. The points in blue are the DSD that was used to fit the model, and the red points are the central composite design that are outside some of these are actually outside of the original definitive screening design points. So that evaluation we saw where the performance of the S-VEM model fit to the DSD is actually being evaluated outside of the DSD design region. So we're seeing greater generalizability with our approach than the standard techniques. And let me see if I can unshare. There's still a couple more questions. Let me see. Someone pointed out that this CCD data, it is a true test set. So there was no, in the JUMP framework for machine learning, the CCD were completely independent of the model fitting and the model valuation during the fitting process. The CCD were completely independent observations. So that's correct. We only have a couple minutes left. Would you like to offer some comments here at the end, Wayne?
Yeah, I was just going to kick in. Thank you for that, Chris and Phil. Thank you both for really a great lecture and a terrific demonstration. Just so that everybody knows, there's a couple of questions, I guess, that do relate. One around the mixture design course that Phil and I are doing. That should start later next month. And you're welcome to email me. A good idea would be, if you're on LinkedIn, just follow Predictum on LinkedIn, because then you'll see that. But if I may, we'll email the folks on this list, if that's okay with you. There's another question about the S-VEM product. It is from Predictum. It's built on JUMP Pro, so you do need JUMP Pro to do it. Now, there is a document, if you don't have JUMP Pro, as to how you can get an evaluation copy of JUMP Pro to use for 30 days or so. So we can, we provide you with that link. You can get that, let us know, and then we can provide you with the S-VEM add-in. I'm just trying to see if there's any other questions here. Oh, as far as the materials, we've kind of put some announcements up through the course. Just email me at Levin@predictum.com, or you could just go info@predictum.com. That's fine. It'll reach me. We'll know what you're talking about. So if you're not able to download the slides or the other materials in the attachments, we'll find another way somehow to get that to you. So I really appreciate the interest, and been in this game for many decades myself, and this is just one of those things that has really, really impressed me. As I said during my bit of the talk, we've been using this ourselves with client data, and it's just been remarkable. So and that's why we produce the add-in. So we want to help make it available for you, and this is a product we're going to keep expanding its capabilities over time. So I think with that, again, I'm going to thank everybody for joining us today, and let's keep the conversation going. So thank you again, Chris and Phil, for all your hard work, your pioneering work this summer, and thank you all for taking the time to join us. Take care. Have a good rest of the day.
Thank you.